All right, time to go teach. Welcome to this next episode of Tutor Tutors, where we are going to look at the method of science. So we're going to look specifically at what process scientists go through to learn about the world. Targets for the day. First off, understanding what a scientific theory is. The goal here is just to make sure that we get rid of a little misconception that there is about what a theory is, what a theory in science is. Okay? So the first goal of the day, or the first target of the day, is to learn what a scientific theory is, which, notice that word, scientific theory. That means it's not the same word that you necessarily hear out and about. So a scientific theory is special because it's not just, as some people refer to, a theory. Okay? Next, our next target is to go through the steps in the scientific method. And lastly, explain the components and the importance of a controlled experiment. And to do that, I'm going to go through a controlled experiment that I did just a little bit ago. First, science. What is science? Well, it's a process. It's not a thing. Science is all about learning about the world around us. It's about studying and understanding why different events or different things take place in the natural world. It is an organized analysis, and it has to be really strictly organized. Otherwise, you cannot prove the cause of an effect. So our goal is to make sure that we can actually prove what it is that we're seeing and why it is that we are seeing what we are seeing. But before we get into that, I want us to understand a little bit about the process of science. The process of science is to understand the world around us. And that process means that we go from the side of being 100% wrong toward being 100% correct. Now, we don't get to 100% correct typically in just one step. Normally, it will take multiple steps to get more and more and more and more correct as we go. Okay? So there is a process of learning that takes place. It's not saying that what we learned, it can just get completely thrown out. No, it's just saying that there is a process that takes place as we progress to get to closer to what is correct. An example of that is, long time ago, we used to think that the world basically was flat. And the idea of a flat Earth doesn't completely, it's not completely horribly wrong. If I look outside and I just look as far as I can see, I don't see any curvature of the Earth. If I walk to my mailbox and walk back, I don't have to worry about the curvature of the Earth. I don't have to worry about anything dealing with that. It's just, for me, it's just basically flat the entire way. So it wasn't 100% wrong for a small area. But we know that is not 100% correct either. We can get more correct because we know that this Earth is not flat. We know that it is more round. And so over time, we figured out that the Earth is a globe shape. Typically, most people refer to it as a sphere. So they look at the Earth and they say, well, we are living on this ball, this sphere in space. But the reality is that that's not even as correct as we know today. It's not that the Earth is just round. It's that the Earth, because it rotates, is actually wider at the equator. It's sort of squashed a little bit. So it's not perfectly round. It is actually sort of pinched in. It's what we call an oblate spheroid. So that gets us more correct. You see, there's a process of knowledge here, getting an exactitude of understanding. And that's what science is about. It's about that progression to get to the most exact understanding of the world. And that's what we're trying to do. So Our process is to take our brain from this mostly incorrect area and work across to much more correct. That's our goal. That is the entire goal of science all the time. And that process we are going to be looking at today. How do scientists go about trying to learn about the world in a precise manner? That takes us to vocab. In science, vocabulary is extremely important. Exact vocabulary means that we can convey an exact message. But the problem is when there is the same vocabulary word used in one area of life and in another area of life, and they have a different meaning. And that can lead to some confusion. The first area for that is theory and theory. So in the world outside of science, a theory is a good guess. It's a speculation. I have a theory that this will be a good year. I have a theory that you're going to enjoy this video series. I have a theory that you will gain a great appreciation of biology. All of those are theories. They are just guesses, and they're just me tossing things out. They might be right, they might be wrong, and they're not based on almost anything. That's not what a scientific theory is. If a scientist says something is a theory, like cell theory, for example, cell theory is not a good guess. Cell theory is not something that's just thrown out there and maybe it's real, maybe it's not. Cell theory is based on science. It's based on understanding. It's based on evidence. A scientific theory is well tested. It's an explanation that explains a broad range of observations, a broad range of hypotheses. It is what enables us to make accurate predictions. A scientific theory is a culmination of research that has gone through multiple steps to finally actually answer a question, to finally be able to explain something that's happening in the world. That is a scientific theory. It's not just some random little guess. It's based on a ton of evidence. It's based on a ton of experimental data. It's based on years and years of work. So when we talk about theory in science, recognize it's not just this word that gets thrown out lightly. It means a lot for something to be a scientific theory. To get there, we need to do the scientific method. That's how we learn about the world around us. That's how we solve our problems. That's how we answer our questions. We have to follow specific steps. Now, these steps that I'm going to give you, I'm just going to tell you there is not a 100% exact step-by-step method. Okay? We typically see it in textbooks, and some textbooks have 7 steps, some textbooks have 6 steps, some have 8. It varies. And no, they don't all have to go in this specific order that I'm about to give. But this is just a general, simplistic overview of the steps that you can take. Okay? Step 1, basically, you observe something, you ask a question. Observations, these always occur through your 5 senses. That's all it is. It's just making an observation, like if you hear a sound or you see a bird fly. But then you ask yourself a question. Why did that bird fly over there? Why did I hear that sound? What was going on? Next, you infer, you use the information that you know already, and then you form a hypothesis. A hypothesis is the answer to the question. It is your best guessed answer to the question that you're posing. So based upon your observation, you ask yourself a question, you then use your background knowledge, and you take a guess. Now that guess can be right or it can be wrong, and it doesn't matter if it's right or wrong. The only thing that matters is you have to be able to test your hypothesis. And that's where we come into designing the experiment. Experimental design is the most important step of the scientific method, period. If you don't design a good experiment, you cannot prove that you understand anything. That's the way it works. So we're going to focus a little bit upon this step and how do you design an experiment? What do you need to have to actually make sure that you are able to prove something one way or the other? Next, you're going to collect and analyze data. Throughout your experiment, you're going to collect some data, so then you're going to analyze it. You're going to try to figure out what it says, what does it mean. Then you work your way across. You're going to make some sort of a conclusion based upon the analysis that you just did. Then you share your results, and probably based upon what you just did, you can ask another question and then continue and cycle around and around and around and around and around, because you probably haven't answered every question in association with that topic doing one experiment. So now we're going to look at a specific, simple experimental design. That's where we're going, designing our experiment. Here's how it works out. Controlled experiments are the main tool that any scientist uses. You have a controlled experiment, the first thing is you're only allowed one manipulated variable. That means that you are only going to have one variable that you control and you change. That's it. If you control more than one variable, you don't know which variable you changed impacted the results. So the cool thing here is, if you only change one variable, that means the impact has to be based upon, or is probably based upon, that one variable that you changed. If there is no change, then you know that that one variable that you changed didn't matter for what it was that you were looking at. All other conditions must be constant. Again, if any of the conditions change, you don't know which condition caused your results. So the whole thing here is to make sure that you know the only thing that is changing. The aspect of the experiment that changes based upon what you changed is called the dependent variable. That is the one thing that's allowed to change, but you're not changing it. It changes as a result of what you changed. And sometimes the dependent variable doesn't change at all, which means that your independent variable did not affect the dependent variable. And that's fine. That means you learned something. You learned that those two conditions are not necessarily related. Then you have to have a control group. The control group is what you are going to compare your experimental group against. And control groups never experience the independent variable. So whatever condition you are adding for the experimental group, you have to make sure that the control group doesn't experience that same thing. But everything else has to be exactly the same. Otherwise, again, you cannot compare them because something else might have caused a change. So the two groups, the experimental group, which is the one that you changed the manipulated variable for, and the control group where you do not manipulate anything, every other condition must stay constant. If those conditions change, then you don't know what you're actually looking at. And then you must collect your data through the experiment, either quantitative data, which would be numeric values, or qualitative data, which would be descriptive words about what has occurred. So I decided to do a little experiment using termites. Termites are little insects that live in the woods, and since I live near the woods, I decided I would go out and I would catch some wild termites. And the reason I decided to do this is because I have heard that termites can be attracted to ink inside of a pen. So I wanted to test to see if this is really true or not. So the first thing I needed to do to actually study this is I had to capture termites. And I did that by going out into the woods, breaking open some logs, and gathering termites. I then put them into a small petri dish. The goal was to transfer them onto a piece of paper. The piece of paper was going to have either a circle that was filled in entirely with pencil, or to place them in the middle of a circle that was filled with first pencil and then secondly ink. The reason that I did both pencil and ink is because, remember, the control group and the experimental group must have all the same conditions, except the experimental group also has the independent variable, the manipulated variable. And so the ink is a manipulated variable. That means I needed to have paper that had just pencil and paper that had pencil and ink. So that way I could make sure that it wasn't the pencil that was actually impacting this whole experiment. The circles were the size of quarters. The termite was placed in the center of the circle, and then it was timed to see how long it took for the termite to walk out of the circle. Once the termite walked out of the circle, time stopped, and we moved on. I repeated this for 5 trials. One part of science is, when you're designing an experiment, you have to make sure that you use multiple trials, because otherwise you don't know if what you actually observed was just some random freak occurrence. If I flip a coin and it comes up heads, that doesn't mean that every time from then on it's going to come up heads. If I flip a coin 2 times and it comes up heads again, that still doesn't mean it will always come up heads. The more trials, the better your data will be, the better your results are, and the more confident you can be of what you actually study and what you actually understand. So here, this is a typical setup where you can see the circle. This is both pencil and ink filled in. And there's our termite right down here. And what you can see is, this is in the middle of the experiment, and it just runs around. And so during this entire time, while it was in the circle, up, and now it left the circle, up, and it's gone. The entire time that it was within the circle, the timer was going. And once it left the circle, the timer stopped. So let's look at the data that we actually got from this. So the data is, we had 5 trials and then calculated the average and standard deviation and standard error of the mean. For the pencil, well, as you can see, I mean, the first trial, honestly, it just walked right out. It spent no time in the center of the circle. I almost didn't have time to actually do the stopwatch. It was so quick. The next few times, it was a little bit longer, and then the last time, it decided to hang out for a couple, little bit of time. But how did that compare to the ink? Well, as it looks, I mean, it stayed in the ink a lot longer. The first one stayed in for 60 seconds. The second termite stayed in for 46.09 seconds. Next, 50.71. It seems like there is a big difference between these two. But in science, it's not enough to just say that there is a big difference. We have to analyze our data and see if there actually is a big difference. And so, charted a little graph over here, just so you can see. So that graph right there, that line, it's very hard to see. That is the average length of time that the termite stayed within the pencil circle. And it's very hard to see, but that little black part right there, that little black mark, that is the error bars. And then this is how long that the termite stayed within the black Bic pen circle. So it stayed, on average, much longer, and there are the error bars for that sample. And what you can see here is, since the error bars don't overlap, we can say that the termites spent a significantly different amount of time within the Bic circle than it spent in the pencil circle. Now, this doesn't have to be where I end. Yes, I've shown that the termites will spend more time within the ink than it will spend within the pencil circle. But is it black ink? Is it Bic ink? What is the reason behind it staying in that specific circle? I don't know. All I know is that in this case, if we do the comparison with the pencil, our control group, or the experimental group of our black Bic pen, it spent a significantly longer amount of time in the black Bic pen circle. And so now I have all these other questions that I can start to ask, and then I can do more experiments to learn more. I have moved a little bit more toward exactness, but I haven't gotten all the way to where I fully understand the answer to this question yet. I've just moved a little bit closer to knowing it. So in summary, we can't understand the world without science. Science is all about understanding the world. Without science, we don't know anything about the world around us. And so it's extremely important for us, as we learn about things, to make sure that we are progressing and growing in every way. So that's one of the huge keys about science. It's about understanding about the world. Next, through science, we're able to grow in knowledge to get more and more and more and more and more correct. And it's not, as I said, it's not a one-step thing. You don't just all of a sudden know everything. The goal is to grow one little bit at a time, and that process, that's science. And a good experimental design, that is crucial when you're trying to explain the phenomena going on around us. If you don't have good experimental design, you cannot explain what is going on. Period. That's it for this time. Be awesome. Stay awesome.